Hello and welcome back. As we continue our discussion of biomolecules, we're going to look at our third group today, the lipids. So we've already looked at carbohydrates and proteins, so let's continue today with the lipids. And as we can see, they're not too much different than the carbohydrates. We're going to talk about hydrogen, carbon, and oxygen. But in this case, the big two are going to be hydrogen and carbon. So there's a lot more hydrogen and carbon than there is oxygen. We talked about the carbohydrates always having a 1 to 2 to 1 ratio. Well, the ratio is going to be a lot less for lipids, so there's very little oxygen, as we'll see in a diagram in just a second. So the basic structure of lipids are there's fatty acids, and they are bound to a molecule called glycerol. So we're going to end up with these things called triglycerides. And so the triglycerides are going to be kind of our basic unit of the lipids that we'll look at. So as we go through the function of lipids, the big one is going to be energy. So there's long-term energy storage, and so we can use that for a variety of activities. Insulation, okay? So if we look at seals and whales and the blubber they have, a lot of animals will use lipids for insulation. It also, the padding around organs, internal organs, to kind of shock absorbers. And then a big one that we'll talk a little bit about now, but when we talk about cell membranes later in the year, we'll talk about biological membranes and how phospholipids are a huge part of that and how they help maintain homeostasis, and really cells and life in general wouldn't be possible without the structure of those. So here is a fatty acid. Now, what makes the fatty acid an acid? Well, right down here on the right, here's our carboxylic group, as we've seen before, right there. And that's really the only place where the oxygen is going to be found, because if you look at the tail, we've got this long hydrocarbon chain, and that's where all the energy is stored. All these carbon-hydrogen bonds, as we break those bonds apart, energy is released that living systems can use. So that's why a fatty acid is so high in energy. Pound for pound, gram for gram, it has more energy than proteins and carbohydrates do. And here's just a quick diagram of the breakdown of it. So lipids are broken into two categories, fatty acids and steroids. Steroids we'll mention briefly at the end, because there's cholesterol and a couple structures, testosterone, estrogen, that look like them. But then fatty acids are going to be the big one we'll concentrate on. And within the four groups there, the two that we'll really look at are going to be the triglycerides, which we've mentioned, and then the phosphoglycerides or phospholipids. Waxes, we'll show a quick diagram at the end. And then sphygmolipids are important because of signal transmission, so with nerve cells and such, but also cell-to-cell recognition. We're not going to mention those a lot right now, but I just want you to know about them. The two, again, that we'll concentrate on are going to be the triglycerides and the phospholipids. So now let's look at fatty acids. And types of fats that you might already have heard about are saturated and unsaturated. So if we look at the top in the upper left here, we've got a saturated fat. And if we kind of ignore the carboxylic group in the red for a second, just look at the hydrocarbon chain. Look between the carbons, and what do you notice? They're all single bonds. Okay, that makes it saturated. It's got the maximum number of hydrogens that it possibly could. Now look just under that, the one here, the unsaturated. Again, ignore the carboxylic group. But notice that right here now, we've got one double bond. When we put that double bond in, each carbon can only have a maximum of 4 bonds around it. So we've got to remove some hydrogens. And what that does is it makes it unsaturated. Now that's going to be important in terms of the health aspect, but also the structure of it, because if we look at the diagram down here, and if we look at the one right here, the saturated fat, right there, hang on a second here, let's see if we can fix this. Sorry about that. Here, there we go. Try this again. So if we look at the saturated fat right there, you can see the 3 tails are all straight. So when there's a lot of them together, they're going to be able to group, to kind of bunch together, and that's going to make it a solid. So saturated fats tend to be solid, whereas if we look over to the right, the unsaturated fats, what happens every time there's a double bond, it puts a kink in the tails. And those molecules aren't going to be able to pack together as well, and they're going to remain a liquid at room temperature. So in terms of the fats that we see and such, hold on a second guys here, my marker seems to be going crazy. Let's try this one more time. There we go. They'll stay liquid at room temperature. So let's look at the good fats and bad fats. So the bad one's over on the right real quick. A lot of you may know this already. So the saturated fats, so fatty meats, chicken skin, butter, cream, a lot of the dairy products. So they are all going to be made up of fatty acids that are saturated. And then in just a minute we're going to talk about these trans fats that you've probably heard a lot about lately in the news and such. And this is really starting to bug me, but we'll continue on. Here we go. And then we got the good fats. So we got the monounsaturated, the polyunsaturated. So these are the ones where they're going to be oil. So you may have heard of olive oil, canola oil. They're the ones that are going to be liquids at room temperature, and they're healthier fats. So the saturated fats, as we said before, lard, butter, animal fats, they're unhealthy for you. They're going to be solids at a room temperature. The unsaturated fats, plant oils, corn oil, olive oil, they're the healthier oils. They're going to be the ones that are liquids at room temperature. So how do they affect cholesterol? So you may or may not have heard there's two types of cholesterol. There is the bad LDL, the low density lipid, and then there's the good high density lipid cholesterols. And the two that I want you to look at real closely here are down at the bottom. We've got the polyunsaturated fats, and they're the good ones because look what they do to the bad cholesterol. They lower it. And at the same time, what do they do to the good cholesterol? They raise it. So we get the double benefit. Now look at the trans fats. They are the exact opposite. They increase the bad cholesterol while decreasing the good. So those are the two that I really want you to look at. And polyunsaturated just because they are the good fats. And trans fats we're going to spend a couple minutes on right now. So if we look on the top diagram here, again, here's a saturated fat, all single bonds between the carbon. We've got oleic acid here in the middle. We've got it's monounsaturated, so you can see there is just that one double bond there in the middle. And then at the bottom we've got linoleic acid that is polyunsaturated. So it's got the two double bonds. So just a quick review here so we know what we're looking for when we look at these bonds between the fatty acids. Now, what can happen is sometimes we can take a good fat, so at the top here, here's a polyunsaturated fat. So that's what we might call a healthy fat. And what will happen is sometimes people don't like to pour butter on their toast, they like to spread it. So I can't believe it's not butter. All of these kind of margarines, what they will do to make them solid is they will hydrogenate them or partially hydrogenate them. And what that means is they undergo a chemical process where they break the double bonds and they add in hydrogen. So they hydrogenate it. So if we look at the bottom here, we've got extra hydrogenation or hydrogens in there. And in some cases, that can make the fat actually more unhealthy. And let's take a look at how that happens. So if we look at the top one here, stearic acid, this is saturated, and that's an unhealthy fat. Now in the middle, we've got an unsaturated fat, linoleic, and that's got a couple double bonds, so that's polyunsaturated, so that's a healthy fat. Now what will happen sometimes is when we hydrogenate one of these bonds, what happens is the way that it bends, it puts it in a formation called trans. Now why that's important, if we look down in the lower right here, we can see the saturated fat is a nice straight line. Now, why is that bad in terms of health? Do you remember? Well, because it's straight, it allows those triglycerides to pack together, which means that it can be solid. And in our arteries, we don't want it solid because that can cause buildup. Now, look at the middle one, the unsaturated fat. Look at that big bend in the tail, the big kink. Well, that makes it one of the healthy fats because they don't pack together real well, and it stays liquid. It stays more mobile. Well, now look at the trans fat. When we hydrogenate it, and we get this little kink in there, what does it more closely resemble? Well, it more closely resembles the straight saturated fat. So we've taken a relatively healthy fat, something that's maybe polyunsaturated, and we've turned it into something that's very unhealthy. So that's why in the last few years you see a lot of marketing towards products that are 0 grams trans fat, because this trans fat is really bad for you because it raises the bad cholesterol and lowers the good cholesterol. So let's look at real quick at the triglyceride. So here is a glycerol molecule, and it's a triglyceride because we've got 3 fatty acids, and they're all linked together. Well, how are they linked together? If you remember, we're going to talk about condensation reactions. So here's our glycerol molecule, a little bit more structure to it. Here is our fatty acid minus the hydrogens, and as you can see, we've got the 2 hydroxyl groups, the OHs, and the black box shows where water can be removed. So if we kind of copy that process 3 times, we get what's shown in the lower right-hand picture here, where we get 3 fatty acids bound to a glycerol, and that's our triglyceride. And here is a prettier version, if you will, again showing a fatty acid, one of them at the top, and water being removed. And then we go through and we get our triglyceride when that happens 3 times. And just as we saw with the carbohydrates, where we called it a glycosidic linkage, and with the proteins, where we called it a peptide bond, when it happens with lipids, we call it an ester linkage. So that's just the special name for a condensation reaction. And here is a picture of the triglyceride, a cartoon version of it, if you will, and with the 3 tails. And one of the types of fats that we talked about that is going to be very important is this idea of a phospholipid. So we've got the 2 fatty acid tails over here, but instead of a third, we've got a phosphate, which makes it polar. It's going to put a charge on it. That's going to be very, very important when we talk about membranes and add that in. So just keep in mind that a phospholipid is the same as triglyceride. It's just had one of the fatty acid tails removed and replaced with a phosphate. Now, I said briefly that we would talk about steroids. And steroids are very closely related to cholesterol. So the diagram in yellow here is actually cholesterol. And you can see it's got this characteristic ring shape where we've got these 4 carbon rings. Now, if we look at the other diagrams, here's cholesterol. So that's what we just looked at. And here is testosterone, cortisol, aldosterone, which are different steroids produced by the body, and they all have the same ring structure. And that's what you need to know about steroids for right now, is that they're all kind of made up the same backbone, the same structure. And we'll talk a little bit more about them in the endocrine system when we get there. And the last thing that we'll talk about real quick are waxes. So here is honeycomb. And here you can see the big thing that I want you to see about here is they've got a very characteristic in the middle of all of them. They all look very similar. They got the double bonded oxygen to a carbon and a single oxygen, but they both have, or they all have, long branches of carbohydrates on either side. So waxes aren't going to be real important in our discussion of lipids as we go through, but I just wanted to see what they look like. So that wraps up our third biomolecule of lipids and leaves us with 1 to go. So in our last session, we'll talk briefly about nucleic acids.